So thank you very much for the introduction. Shall I go ahead and share my screen now?
Sure.
Yeah, okay. So let me... Okay, is that working in its full screen for you? Yep, okay, cool. Let me just make sure I get the chat window up here too in case... Oh, of course that's not working. In case there's any questions or anything. Okay, so I have the chat window up here too, just... Okay, so thanks very much for the introduction, and I'm really happy to be here and give this talk. So all of the talks up till now, and hopefully this one as well, have been really great in the series, and I've learned so much. And so I'd like to thank Vivian for just introducing me and the previous speakers, and especially James Kroll, who really gave like an awesome introduction to I think the kind of stuff I'm going to talk about now. So I'm going to talk about molecular and neural circuit effects of insecticides and implications for pest control and pollination. And I think that's a title that somehow got generated for my talk. So I generated a second title, if my mouse works. Okay, and my second title is How Can We Use Drosophila to Inform Our Understanding of Pesticide Function? And before I start going in that direction, let me just say one thing, which was already hinted at a little bit. So this talk is the first time I've talked about a lot of this. And so if there's stuff that's totally unclear, it's not your fault, it's probably my fault. So if you need some clarification to go on with the talk, please do interrupt me. And in general, offline or afterwards, I welcome any comments that could improve our work in the future. Okay, so I want to start with the hypothesis that we could use Drosophila to understand the molecular and physiological mechanisms of pesticide effects across insects. And so, you may know of Drosophila as an organism whose natural habitat looks something like this in the basement of my building. Of course, that's a joke. It's not really their natural habitat. But the question is, what can we learn from this lab animal that might be relevant for understanding the effects of pesticides in nature? And, the first thing I want to address is why am I even talking about Drosophila? And so I think it's a wonderful system for addressing many questions in biology. So flies have been there since the beginning of genetic research. In fact, they were some of the key organisms in understanding the nature of inheritance in the gene. And so we have many, many tools and vast knowledge from this more than 100 years of genetic research. To work on Drosophila, you only need very modest things. You need only a small amount of space, modest equipment, modest materials, and a modest amount of money. And because there's so much work on Drosophila going on at all kinds of different levels of biology, molecular, cellular, developmental, neurobiology, and probably a bunch of other kinds of biology, like let's say ecology, that I'm forgetting, that I have forgotten to put in this list here, it's just a great system to work in because there's all these cross-links that you can make in other dimensions. There's many Nobel Prizes which have been awarded for work on Drosophila. It was the first sequenced animal genome, and it's just a great system. And fundamentally, I think for me, the great thing is that it's relatively easily to traverse biological scales with Drosophila down to the molecular level. And one of the major reasons is here, this image at the bottom of the screen, the Bloomington Drosophila Stock Center, which is at Indiana University, and in fact descends from original stock collections from Thomas Hunt Morgan 100 years and more ago. And at the end of last year, I just found this on their website, they had over 76,000 different stocks that you can literally just order. And to be honest, I don't even know how much the stocks cost because by far my major price when I import them is the FedEx shipping and the border veterinarian fees. The the actual cost of those is relatively low. And there's just an unbelievable richness of genetic tools that we have there, which I unfortunately don't have any time to go into at all today, but it's a remarkable system to work in. Okay, so back to this question. So you could say, okay, but the diversity of insects is extreme. So why should we? What's going to be useful about Drosophila to understand effects of insecticides or other pesticides in the wild? And I would say, well, okay, but evolutionary conservation of molecules and biochemical pathways and neural circuits and behavior, that might suggest we can use Drosophila to inform our understanding of other insects. And I will show you a little bit, some slides about that in a minute. And furthermore, it makes commercial sense that insecticides are developed to be broad spectrum and to target a wide range of insect species. So for example, I've heard a scientist from biocrop science saying very, very specifically that this is the goal when they develop an insecticide, because it costs a huge amount of money to bring one of these to the market, and so they would like for it to be useful against a huge range of insects. So in fact, I think there's even a commercial desire that these insecticides would work across insects, and therefore it would make sense that to some degree at least, we could use a lab animal like Drosophila to study the effects, and that might be relevant for other species as well. Okay, so to this question of evolution, I wanted to show this slide here from Strausfeld, Ma, and Edgecombe from a few years ago, which shows a region of the insect brain and insect relatives called the central complex. And so here is a family tree. So the chelicerates like spiders, Myriapoda, the myriapods like millipedes, Pancrustacea, which includes as a subgroup within the Pancrustacea the hexapods, so the insects. So all of these arthropods, or all of these organisms, have this brain region, the central complex, where a lot of the neural structures you can clearly see all the way back into deep evolutionary time, this conserved kind of brain structure, which is right in the middle of the brain and is thought to coordinate movement in the animal. Okay, so the main point here is just a very simple one, that brain structures in the Euarthropoda are ancient, and in fact even older than the insects. They predate the splitting off of the insects from the crustaceans and the other sister groups. And when you look at different species of insects, even from different orders of insects, you can see that the major brain structures are obvious, and they're homologous in these different species. So for example, here's Drosophila, and in blue are the antennal lobes, where the antennae send their projections, and this is a olfactory processing region here. And you see that in all these different species. You see the medulla in yellow. This is part of the optic lobes that the fly sees with. The lobula complex, which is one synapse deeper, one synapse closer to the central brain from the medulla, and so this is doing higher level visual processing. The mushroom bodies in this pink color, and you see these are massive in the honeybee here. These mushroom bodies are very important for learning and memory, and bees are amazing animals at those kind of tasks. And then in green is this central complex region, which I had just mentioned. And so even across very evolutionarily distant insects like the locust, these structures are very clear to see in these different animals. So there's a huge degree of evolutionary conservation, at least at the macro scale in the insect brain, if you call the insect brain macro scale. But if you go to the molecular scale, it's also similar. So for example, here is a table showing the percentage identity between Apis mellifera, the honeybee, and Drosophila melanogaster nicotinic acetylcholine receptor subunit protein sequences. And so talk about these a little bit more in a minute, but these, the nicotinic acetylcholine receptor subunits, these are the targets of the neonicotinoid insecticides and other insecticides as well. And so you see that between Drosophila alpha-1 subunit and Apis mellifera alpha-1 subunit, 71 of the 77 amino acids are conserved, and that's a higher number than the conservation to any of the other subunits of those species. And it's the same, so you can identify basically the corresponding subunits across these very, or potentially very divergent insect species. And so you can, using this type of data, these authors have made a phylogenetic tree of the different nicotinic receptor subunits, and you can see, for example, the Drosophila alpha-1 subunit group is most closely related to the, this is a mosquito, Anopheles gambiae alpha-1, and the Apis mellifera alpha-1. These are more closely related to each other than they are to other nicotinic subunit genes. Okay, so there seems to be a high degree of conservation also at the molecular level, which would suggest indeed that looking at these, looking at Drosophila may be useful for understanding insecticide effects across insects. Okay, so let's look a little bit deeper at this nicotinic receptor. So I think nobody has talked too much about this yet, so I thought I would spend 1 or 2 minutes discussing these. So the nicotinic acetylcholine receptor is sitting in the membrane, typically of a neuron. And the idea is there are some positively charged ions outside of the cell. And normally this thing would be closed. But when acetylcholine, which is the dominant neurotransmitter, the dominant excitatory neurotransmitter in insects, when acetylcholine binds to this channel, it opens, and these positive ions, for example, calcium, can flow into the cell. And so then they depolarize the neuron and it can fire. And like I said, the acetylcholine is the dominant excitatory neurotransmitter in insects. And these nicotinic acetylcholine receptors are, those are the class of ligand-gated ion channels which acetylcholine binds. Okay, so they're pentameric. That means each ion channel is composed of 5 of these different subunits, and typically they're heteromers. So you can have alpha subunits and beta subunits. And in fact, depending on the composition, so you've got to have 5, but depending on which exact ones, if you have like alpha-1, alpha-2, or if you have alpha-1 or alpha-2 or alpha-3, all the way up to alpha-7, the properties of that ion channel can be quite different. So for example, different insecticides bind to different subunits with greater or lesser affinity. And in fact, to make that more complex, that also depends on the specific insect species. But I think about this, I think about the result of this, that the different subunits mean different properties. I think about that in the way that the acetylcholine system isn't exactly just one neurotransmitter system. Because it has this diversity of receptors, I think of it actually as many different types of neurotransmitter systems. At least on the receptor side, these can have quite distinct properties. So the neonics and other insecticides like the newer sulfoxamine, for example, these bind this acetylcholine receptor at the acetylcholine binding site. And one of the papers which I asked you to read had this figure in figure 1. I thought was a great demonstration of the effect of these neonicotinoids. So the idea here, this is recording over time doing calcium imaging. So this is the y-axis is essentially the concentration of calcium. And at time point 1 minute, they introduced a low concentration of imidacloprid, as we've heard already, one of the most dominantly used insecticides and an agonist for the acetylcholine channel. And so when they introduced a little bit of imidacloprid, you see this immediate influx of calcium to the cell, and this low level of calcium is maintained. And then in comparison to a control animal where no imidacloprid had been introduced, when they spritz on a tiny amount of carbachol, which is an acetylcholine substitute, it's chemically similar or identical to acetylcholine, the neuron responds strongly in the control case, but in the case of the imidacloprid, the response is strongly attenuated. So this is called, imidacloprid is called a competitive agonist for the nicotinic receptor because it's binding in the same place as acetylcholine. So it would block the normal action of acetylcholine on that cell. And you could say, well, okay, but all this calcium has been accumulating for 5 minutes here, so maybe the intracellular stores of calcium are just depleted, and so that could be some aspect of the. So then the authors used a different drug, thepsigargan, which releases calcium from intracellular stores, and there you see a very strong response in both conditions, suggesting that the imidacloprid is not affecting the intracellular calcium stores, but rather blocking the entrance of calcium when acetylcholine or carbachol would be applied. Okay, so let's say a few more words about these subunits. So if we kind of zoom into a cartoon drawing here, you see there's 4 transmembrane domains, and I've tried to draw those in this channel, in the complete picture of the channel here. And in Drosophila, there are 7 alpha subunits, 3 beta subunits, and in Drosophila, the lab of Trent Perry has shown that if they make a knockout, so they make a fly with no alpha-1 subunit in its genome, these flies are largely resistant to imidacloprid. So that suggests that the alpha-1 subunit in Drosophila is the main target of imidacloprid. Okay, and of course, these channels are on the dendrites of neurons. They're on the postsynaptic side, and so acetylcholine would be secreted into the synaptic cleft here and normally would activate these channels and trigger a response in a downstream neuron. And so, the big question would be, so how are insecticides affecting neural circuits where insecticides have been applied? So we want to address especially the sublethal effects of insecticides, and that's what I'm going to focus on here. So I want to say a few words about sublethal effects, and so I'm sorry for this kind of wall of text, but I want to say that lethal dose and lethal concentration studies, they're done in highly reproducible but highly unnatural conditions. And these sublethal effects, when they're measured in more natural conditions, they show the effects of insecticide at low doses can lead to decreased bee and other insect health. And so given this evolutionary conservation of these nervous systems, could we expect similar effects on the health of less studied insects? And so field studies, of course, are the ultimate in realistic, but they're very difficult and expensive. And so the idea why to look at Drosophila may be that by doing laboratory studies where we can look at mechanisms of pesticide effects, we can learn things about them that could allow us to predict the effects of pesticides more generally and with real-world relevance. Okay, so I'm going to now get a little bit more specifically into the kind of research which my lab does. So I'm going to address the specific question of how does sublethal imidacloprid affect vision and visual behavior in flies. So this is beautiful Aristalis tenax, a hoverfly. It's actually the species I did my PhD on, also a pollinator. And here's a movie of Aristalis taken by David Attenborough or his team back in 2005. It's still an amazing video. It's probably, I went over this with Natalie. I can play it, I think, a little bit better if I do what I just did. So this animal uses its vision as its primary sense of how to move throughout its world. It uses it for its sense of how it's moving relative to the world. And so in order to stabilize this beautiful hovering that these hoverflies do, they're very highly visual animals. And my research over the last 20 years has really been focusing on vision in flies. And I'll talk a little bit about that today. Before I talk any further about that, I want to say in fact, the example that really got me into insect vision in the very beginning, which is the fact that insects can use vision to find home. And this is very common in Hymenoptera. And so here's a picture of a wasp, the bee hunter, Philanthus triangulum. And they dig their nests, the females dig their nests in sand dunes, a little hole. They lay their eggs there, and then they go out, typically flying, and catch a bee. So that's why they're called bee hunters. They typically catch, for example, Apis mellifera, bring them back into their hole, and then provision their larvae with this as food. And already, almost 100 years ago, Nico Tinbergen and colleagues did these simple but elegant experiments where they would surround a nest hole. Oh, let me switch off my... Okay, so they would surround a nest hole with shapes, and when the female wasp would fly out, she would turn back and look at these shapes and make these arcing flights as she would leave her nest. And then they would play this kind of trick on her. They would move these shapes to the side, and when the wasp came back, they could see she would go to those shapes again and start looking for her nest in the middle of the ring. And so with this simple experiment, they demonstrated that these wasps can use vision to return home again, and that they use vision also as a dominant cue compared to, for example, olfactory cues. And so I just think this is absolutely fascinating, that this tiny animal has this capability of remembering what its home looks like and how to get back there. So how do insecticides affect insect vision? I'm going to start with a recently published paper from Parkinson, Zhang, and Gray, where they were, first they measured mortality as a function of insecticide concentration. I'm going to focus on the imidacloprid here, which is the purple dots. So they found lethality at concentration here, and in control animals, so they're testing locusts. In control animals, they were showing there's a black dot, and I don't know if you're going to be able to see it in your movie, but it suddenly expands. It's this rapid expansion, and the locust thinks, okay, something's coming at me, so I got to jump and get out of there. It jumps and leaves. And in the imidacloprid-treated animals, the expanding dot looks the same, but the locust just doesn't move for that demonstration. Okay, so they quantify this, and they see that at concentrations at least one order of magnitude smaller than the LD50 for imidacloprid, these animals just don't respond anymore to this looming expansion stimulus. So this imidacloprid seems to have some kind of negative effect on the visual behavioral responses of the locust. So this, so then they did electrophysiology. So they're recording from multiple neurons simultaneously, and they could see, for example, that the response to this expanding stimulus. So this is over time, this stimulus is coming at the locust, and these locusts treated with imidacloprid have a reduced motion response. Okay. So they have this reduced response to visual expansion, and one question could be, so is this effect limited to locusts and visual looming, or could vision be impaired maybe in other ways or similar ways in other insects? Okay, so here is where our virtual reality system comes in, and I think you all read this paper that we published a couple years ago now, so I'm going to show you a movie of this, and let me switch on my, I switched off my screen sharing rather than switching on my optimize for video. Okay, so I hope you can see this video. There's a little red dot here which is following a Drosophila, which is flying in a 1 meter diameter arena. And we're tracking these flies with this multi-camera system. And we have computer projectors shining around the outside of this arena so that, and then what we do in our computer, we have literally computer gaming hardware and software that we render this kind of visual world in. And then we take that visual world and we figure out what we need to show on the walls of the actual arena so that for the fly, when the fly is here, that it would see this kind of visual world. So it's really, if you know the holodeck in Star Trek, it's really, you know, that's a very good kind of mental imagination of what this is. Okay, so if we fly flies in this kind of configuration where there's this central post that we create with our virtual reality inside of this cylindrical arena, and let me now switch off this optimize for screen sharing thing. Okay, so if we now look at top views of trajectories where we have this post, this virtual post in the middle here, we see flies avoid that region. And we don't think that's some kind of artifact of bad tracking there or flies liking to fly in circles or something like that, because if we remove this from our computer graphics simulation, the flies fly right through there. So we believe that the flies really just see this post, and so that's why we think this word virtual reality is a good word to describe what it is we've created here for the flies. And so just one more quick word about this. The basic principle of this actually works for many different species of animals. So here we have it drawn with a fish, and in our paper we publish it with fish. But the basic principle is geometrically quite simple. So the idea is we know where the animal's eyes are, and we know where our arena is. And as the animal moves along, we just maintain the relation between the pixels we draw and the part of the retina that would have seen these shapes. So it requires us to track the animal with very low latency, and it requires us to draw our computer graphics with very low latency. But once those things are done, the rest is relatively straightforward. And so the key idea here is that it's perspective correct rendering for a freely moving observer. Okay, so now, if I can optimize my screen for sharing the video clip. So we can draw not just stationary scenes and embed the fly in this kind of stationary world, but we can create a reactive world where we have a computer program that's running and updating what the fly sees as the fly is flying along. And so in a very kind of simple version of this, we can take advantage of the so-called optomotor response of the fly, and we can use that to redirect the fly along a path that we want. So we have predefined an infinity symbol, and we've said, okay, we want the fly to keep flying along this infinity symbol, and whenever it deviates from that path, we show this visual motion to it to kind of push it back on the path. And so we can get, I mean, this is a like best case kind of scenario here, but we can get data where the animal just goes again and again and again on this kind of trajectory. And so in terms of like the neurobiology of these kind of responses, this is actually nothing novel. We scientists have known that these optomotor responses are a really important component of visual behaviors in flies for decades. But the cool thing is that we have an instrument now that lets us just reliably elicit these behaviors in a freely moving animal in a totally automated and high throughput kind of way. And so this is where Julia Gaburo got excited and wrote me an email and said, hey, I'd love to join your lab and do some interesting science and maybe work on the effect of pesticides in using Drosophila as a model. And so what we have now is we have 10 arenas, so not this 1 meter diameter thing, which is very large, but much smaller, made out of the standard computer monitor size. And we have 10 of these VR arenas, and you see the kind of figure 8 trajectories may not be quite as good, but they're still totally sufficient to do a lot of experiments. So I'm now going to show you some experiments that Julie's been doing, and now this is really the first time I've presented these results in public. And so the idea of her experiments has been that at 10 a.m. in the morning, she will select flies, so we work on female flies, and she will start starving them, so take them out of their food in their vial. So they have several hours of starvation. And then we treat them with an imidacloprid in acetone solution. And we use a method where imidacloprid is dissolved in this tiny drop of acetone, and in this way we can be very sure about the exact dosage that each fly gets. And so we apply this with a microinjector. We don't actually inject the fly with it, but this acetone is absorbed directly into the cuticle of the fly. And so this is how we treat the flies. And so we use several different dosages. So we've tested that, we've tested our lethal concentration 50 with this type of application approach. And over a 24-hour test period, we have much higher concentrations than this, being the LC50. So this value of 1.8 nanograms is our highest concentration. This is less than 3% of our LC50. So we treat them starting at 4 p.m. and starting at 3:30 p.m. and then immediately after we put them into these arenas that we call the fly cubes, and we basically run them in a kind of stimulus paradigm like I just showed you for 19 hours overnight, including their circadian night. And then the next morning we stop the recording, we upload to the data pipeline, and we gather lots and lots and lots and lots of trajectories under lots of different kind of conditions. And so we've been doing two different kind of visual stimulus conditions. One has been what I showed you the movie of a moment ago, the translation condition. So now the idea is that the fly is flying around in this point cloud, and these points are translating past the fly. So if the fly is here and we want it to go here, there's this error vector here. And so we translate these points with a velocity proportional to that error vector. And once the fly gets, so if the fly reacts properly, it gets closer to that point, and then we advance the location of the point and we loop this cycle along. And so this is how we keep the fly on this infinity trajectory. So this is for translational optic flow, like what happens if you're moving, translating through the environment, if you're moving linearly through the environment. So rotation is what happens if you're turning. And so we do something very analogous. If the fly is here and it's flown in from here and we want it to go there, we know that we need to make it turn left. So we move the angular velocity of all these points by some value proportional to this error angle. And then if the fly turns, it gets closer to that point. And then if the fly gets close enough, we advance this target point. And so again, in this approach, we can get the flies to follow this kind of pathway. So we're testing two different kinds of visual stimuli here, a rotational optic flow and a translational optic flow. And both of those are known to be important for insects to stabilize their flight and to fly in the direction that they want to go. Okay, so if we just look at the raw trajectories, maybe you can see already with a little bit of imidacloprid the performance in following this infinity symbol stimulus is reduced. And this is the same both for rotation and translational optic flow. If we look at the entire 19-hour duration of our recording, we can see that maybe more prominently in these kind of heat maps. And one of the things you may be able to see is that there's actually less. So these are normalized, that 1.0 is always the maximum amount for that particular plot, but there's actually less data in some of these. So one of our main effects is that the imidacloprid actually just reduces the flight frequency of these flies. But then if we zoom in and look at smaller analysis windows, so for example, the first few hours, which may help us look more at the acute effects of imidacloprid, just the very immediate effects, we see this strong kind of decreased response decreased flight probability and reduced ability to fly well, both for the rotational and the translational stimuli. But actually with the rotational stimulus, a lot of the ability is kind of recovered after almost a day here, whereas with the translational stimulus, it's reduced. And so one of the things that takes, well, no, let me summarize here. Okay, so we've got this decreased flight frequency, especially in the first hours. We have decreased optomotor performance, and we have lots of data. So I will not walk you through all the stuff that we're still very much in the middle of processing, but I want to show you just one result here at the end of this period from hours 12 to 17. If we look at the distance, the mean distance of the fly to the perfect infinity symbol, these are histograms for the rotational optic flow and the translational optic flow. We see that only for the translational optic flow do the flies seem to have a reduced response to this stimulus with this behavioral measure. And so therefore we think that that imidacloprid reduces behavioral response specifically to translational optic flow at this kind of longer time point. So this is still very much the beginning of this kind of work for us in our lab. But what I hope you can see and what I hope you agree with is that this visual virtual reality may be suitable for testing insecticide effects on Drosophila. It's a high throughput apparatus, it's highly quantitative, and can give us multidimensional behavioral measures. And it is specific enough to allow us to find effects really in very specific kind of behavioral modulations. So now the kind of thing we'd like to be able to do is to use a lot of these genetic tools to investigate the molecular neural circuit and behavioral mechanisms of these insecticides. And as you know, we expect that to be relevant for other species. Okay, so just to give a hint of that, one slide from some work from some other labs, very beautiful work, where I just want to show that Drosophila is a good system to investigate the biophysical basis of visual neuronal computation. So here's a paper from the lab of Axel Borst that just came out. And what they're doing is they're using a transgenic animal expressing the GFP, the green fluorescent protein, tagged onto the Drosophila alpha-7 nicotinic subunit. And what they show in their paper is that these nicotinic receptors aren't just all over the dendrites of these neurons, which are important for motion detection, visual motion detection in the optic lobes of the fly, but they have a very specific distribution in only a part of the dendritic tree. And the authors argue that that is very important for this actual motion detection computation operation. Unfortunately, I don't have any more time to go into that work. I wanted to highlight another application of Drosophila to understanding motion circuitry from the lab of Marion Zillis. And here her lab has done, for example, pharmacology where they specifically inhibit one particular ion channel and measure specific physiological changes associated with that. But then they also use genetic tools, interfering RNA, and knock down that specific channel and measure similar effects. And now the difference here is this effect is measured by knocking it down only in a single neuron type. They're using a transgenic animal to express this RNA interference system only in this one particular neuron. And so they can really get to very, very precise effects really at the level of the individual cell, the individual ion channel. So I think it's a really powerful system. Okay, so I would say, a giant question is, what are the mechanisms of the long-term or the chronic sublethal effects of insecticides? And I think this is a huge challenge, even using the genetic tools of Drosophila. And that's why I also suggested to have a look at this paper, which I think is a really, really great example of using Drosophila to investigate some of these mechanisms. So I certainly don't have time to go through all of this. It's quite a dense and exciting paper. But they, again, tested concentrations of imidacloprid with their application method and found concentrations which were not lethal. So over 8 days, the concentration that they used did not cause a significant decrease in lifespan of these flies, or in mortality of these flies. But it did cause a reduction in movement in these were larval experiments. So at this sublethal dose of imidacloprid, they used a fluorescent dye which shows the levels of superoxide, which is a reactive oxygen species. And what they showed is that after 1 hour of exposure to this concentration of imidacloprid, the levels of the superoxide are increased in the brain, in the nervous system, but not increased in the anterior midgut. Whereas after 2 hours, those levels came up also in the anterior midgut. So the idea is that the brain may be the first place where these reactive oxygen species really are occurring, which might make sense because that's where these nicotinic acetylcholine receptors are. And then they noticed that in a region of the fly called the fat body, when they treated with imidacloprid, the area covered by these lipid droplets increased, and there were more smaller droplets, and the area was larger. And I think what was really cool about their paper is then, okay, so sublethal imidacloprid also increases the area of lipid droplets in the fast body. So then what they could do using Drosophila genetics, again using RNA interference under the control of a specific promoter that would only express this in neurons, they could show that by interfering with mitochondrial function, so knocking down these genes, mitochondrial complex 1 and mitofusin, if they knock those down only in neurons, they could see a very similar effect. So this is a phenocopy of the effect of this effect of imidacloprid by knocking down these mitochondrial neurons, by knocking down these mitochondrial genes in the neurons. And then, based on this kind of knowledge, they did something which I found quite impressive. So they used a drug called NACA, which is an antioxidant, and this was actually the figure I just showed you before. But if they exposed the flies to this NACA antioxidant, they could see that that this area covered by lipid droplets, the effect of imidacloprid, would be rescued by giving the flies this NACA antioxidant compound. And so the idea is that calcium-induced neuronal reactive oxygen species are the trigger for the myriad chronic effects throughout the body of these insecticides. And then that's actually very, very interesting now to think about what that, a couple recent papers. So for example, here's a paper, imidacloprid-induced oxidative stress in honeybees and the antioxidant action of caffeine. So maybe there's an ability to really link these molecular mechanisms from Drosophila to bees. Or here's another paper. Chronic exposure to neonicotinoids increases neuronal vulnerability to mitochondrial dysfunction in the bumblebee. Okay, so really maybe there's something here. And so this paper just came out. There's a huge amount of genetic data in there, which is basically waiting to be investigated deeper, I would say. So it's a very interesting resource. Okay. So before I end, I just want to show a couple things that are, hopefully going to be related at some point. But right now, they're just technologies in development. So I want to show an insect tracking system that we call the millisecond insect tracking system because it operates with very low latencies. And right now, this works indoors in the lab, but we're working on trying to get this to work outdoors. And so here's a still image where Tang, the postdoc who's done all this work, is brushing a locust, causing it to jump. And now what we're going to see, so you see the locust jumping with this just wide field camera. But now this is the view through our tracking system. And hopefully what you notice is that this video looks kind of weird because as the locust moves, including jumping, which is an incredibly strong acceleration, it remains just locked in the field of view of the camera. And I'm really excited about this because it means that we can eliminate motion blur when an animal is moving through the field of view of a camera. Even if you have a really high resolution camera, let me see if I can do the share. Okay, so even if you have a really high resolution camera, that doesn't help you get away from the problem of motion blur. So the nice thing about this system is that because it actually tracks the insect with the entire optical axis of the camera, it keeps it in sharp focus even as it's moving along. And so we've also done this, we've tracked a beetle with this. And the basic principle is that we have a retroreflector on the animal and that we use this retroreflector to stabilize this high-speed mirror-based system to keep the insect directly in the field of view of the camera. Okay, so that's kind of one sort of high-end tracking system we're working on. But we're also working on a low-cost nest entrance and exit monitor for species like bombus or osmia. And so this work is also fun, and there's a lot of, there are a number of other groups making similar kind of tracking systems, but I think they could be hugely useful, and so we're also working on stuff like that. So let me now stop my optimized video, and I just want to end my talk by thanking everyone in the lab. So I've only talked about a few of the projects in the lab. We're doing various other stuff as well. But Julie Gaburo, who's here, she's been the driving force behind all this imidacloprid testing in the fly cubes. And then Tong is the one who's developed this millisecond insect tracking system. And I want to thank all the funding sources we've had over the years, especially the Humboldt Stiftung for funding this work with Julie. And stay safe out there, everyone. And I'd be happy to take any questions, and also after these breakout groups. Okay, so thanks everyone.
So I have a question maybe to get us started. So for your virtual reality system, sort of how many like different sizes and scales of insects could you sort of include in that without having to, dramatically alter the way it's set up right now?
So the big one that's 1 meter diameter, I think that should be totally fine to fly bigger insects like bombus, apis, and maybe even some hawk moths would be extremely exciting. We just haven't. So okay, so the challenge is that as the insect gets bigger, typically they fly faster, but our system doesn't work any faster just because the insect is bigger. So we have sort of, although we've done everything we can to make the reaction time of our system as low as possible, there's a certain point where we can't, if it's flying fast in a certain direction, we won't, no matter what we show it, we won't be able to turn it around in time. And then even if we could, we may get to more or less and less realistic kind of scenarios where all we can do is basically trap the insect between something that's really going to threaten it with like imminent crash. But it would be very exciting to put some bigger insects in there and see how we go. Yeah.
Have you ever coupled the visual cues with some other like olfactory cues or light or?
We would love to do that. It's a little challenging. So when I was a postdoc with Michael Dickinson, this was when I invented the sort of version 1.0 of this. And the software has gotten much more sophisticated, but back in the early days, we were combining wind stimuli with airflow stimuli. So for example, Sawyer Fuller, who I was mentioning before the talk started, he actually, some of his PhD work was looking at the multimodal integration of airflow detection on the antennae and visual detection with the eyes. And those are exciting topics. Of course, so in more recent work, actually, Flores has shown, for example, that olfactory cues cause insects like mosquitoes, appetitive olfactory cues cause them to lock onto salient visual targets. So yeah, there's probably some There's definitely some very interesting things to be done there, but just technically, as you can imagine, having controlled or even, semi-controlled airflow in there, it just increases the technical complexity. And right now, part of what we're trying to do is just do some science now with the level of technical complexity we've got.
Cool.
All right. Well, I don't see any questions coming into the chat. So I think that this is a good time for us to wrap up, and we'll move the students and the rest of the class over to a private discussion now to continue our conversation with Dr. Straw. Thank you so much for everybody who was able to join us. And we don't have a seminar next week, but if you just sit tight, we'll be able to resume again after US Thanksgiving happens. Anyway, thanks so much, and I'll catch you all in the discussion room.